Okay, so now we'll continue with Frederik Gade from Denmark.
Hello everyone, my name is Frederik, and I'm going to present my project about personalizing medicine for treating genetic disorders. And looking at my project is a case about a specific genetic disorder called familial hypercholesterolemia, which is quite a mouthful, but I'll explain later. So first and foremost, it's very important to understand what a genetic disease actually is. And a genetic disease is a disease that's caused by a mutation. And you probably all have heard about mutations as changes in DNA sequence, where one amino acid of the DNA sequence gets changed, or even more amino acids get changed with other amino acids. And this in turn creates, or like base pairs are changed, and the protein resulting from those changes in DNA are changed as well in the amino acids. And from that, you can see that the amino acids actually change the protein structure, because those squiggly lines you see on the screen are a result of the amino acids that the protein consists of. And when you change the amino acids that the protein actually consists of, you actually also get a different fold. So those squiggly lines look pretty confusing. So how do we actually get those squiggly lines? And the answer is you can either find it experimentally or you can use computers. So because I'm kind of a computer geek, I guess, I thought, well, is there any way of doing this? And you can algorithmically optimize the structure. So looking at an amino acid sequence, you can take fragments of that sequence and try and fold it in a specific way and find the most energetically optimal structure. And this is very useful because protein structure actually determines most of the protein's functions in the cell, because the protein interactions between different proteins are things that actually determine much of the functionality within the cell. So this is very important for my project, where I look at familial hypercholesterolemia. Familial hypercholesterolemia, or just FH, is a genetic disease which primarily is caused by mutations on LDLR, which is the blue protein you see on the screen, or the blue polypeptide you see on the screen. And actually this protein is a receptor protein which takes LDL cholesterol, or the bad cholesterol, I guess, from the blood into the cell and destroys it or breaks it down. So if there's a problem with this receptor, you actually can't clean the blood from LDL cholesterol, and you might get the increased risk of cardiovascular disease or even arteriosclerosis. So what I looked after is different mutations, and the red structure you see on the screen is actually a mutated structure superimposed on the old structure. So even a slight small mutation within the protein actually changes the angle and the structure of the rest of the protein quite a lot. And by looking at those differences in the protein structure, you can also examine how the interactions differ from the old structure to the new structure. This creates changes within the pathways in the body. This makes it so that LDLR doesn't bind to the same things the way it did before. So especially for this one, the red protein, there's an inhibitor that normally binds to the blue protein and inhibits cholesterol from being pulled into the blood. Well, this one actually has a better binding affinity to the red one, which in turn explains the disease. This is kind of a way of explaining how the disease actually works, the method of how it actually works. And by targeting very specific pathways in this system, you can exploit that and target very specific mutations in different ways, even though it's the same disease. But it's not really the same disease, because even though the different mutations, they have the same symptoms, and the symptoms are actually what classifies the disease. And this poses also a question, like how can we actually make sure that simulations are close enough? Because they are cheaper than experimental methods, because experiments take a lot of time, they take a lot of work, but simulations can work on their own in the background. But normally they're actually quite precise. You can actually show that in most cases it actually is within the normal range of an experimental method of uncertainty. Even though it actually is based on experimental data in many cases, the difference should still be quite all right. But of course it's not real life, so it's not as certain that in silico methods, computational methods, are as precise as the ones you find in the lab. So even though they are computationally heavy, they are efficient. So supercomputers or even like better computers can do small simulations on their own. And that is my project. Thank you very much.
Thank you very much. Okay, do we have any question? Okay, it seemed crystal clear then. Okay, then thank you very much again.